And so with that, we will start this session by introducing the first speaker, Roger Barker, who cannot be here with us, but we're going to hear his recorded talk. Roger Barker is a professor of clinical neuroscience and honorary consultant in neurology at the University of Cambridge and the Addenbrooke's Hospital. He's had his training both in Oxford and Cambridge and London, and he's a real clinician expert in the fields of both Parkinson's disease and Huntington's disease, for which he's working on developing both cell and gene-based therapies. So with that, I welcome now Roger Barker with his talk.
Thank you very much for asking me to speak at on sleep and metabolism in Huntington's disease. I'm sorry I can't be there in person to do this live, but I hope you find this talk interesting. What I'm going to do is take you a little bit through some of the work we've done in years gone by and update you a bit as to where we're going next with this particular area of work. So to begin with, I thought it would be useful just to sort of rehearse, if you like, the well-known recognized features that we all understand by Huntington's disease. So there are motor problems, the typical early features being the flick of movements, the chorea, the impersistence with keeping the tongue out, the abnormal eye movements, and those are the common early motor features that you see. Coupled to that, you often see cognitive problems around planning and organizing things, very frontal type executive dysfunction, and obviously with time that becomes more prominent. And then many of you will be aware of the fact that in the early stages psychiatric features can be dominant: low mood, irritability, anxiety. Patients in clinic, that some of the early problems came to my attention, or our attention, around that. Thank you very much for asking me to speak at on sleep and metabolism in Huntington's disease. I'm sorry I can't be there in person to do this live, but I hope you find this talk interesting. What I'm going to do is take you a little bit through some of the work we've done in years gone by and update you a bit as to where we're going next with this particular area of work. So to begin with, I thought it would be useful just to sort of rehearse, if you like, the well-known recognized features that we all understand by Huntington's disease. So there are motor problems, the typical early features being the flick of movements, the chorea, the impersistence with keeping the tongue out, the abnormal eye movements, and those are the common early motor features that you see. Coupled to that, you often see cognitive problems around planning and organizing things, very frontal type executive dysfunction, and obviously with time that becomes more prominent. And then many of you will be aware of the fact that in the early stages psychiatric features can be dominant: low mood, irritability, anxiety, apathy, and such like. So whilst we recognize all of those features, I thought what was interesting for the talk today would be that it was through seeing patients in clinic that some of the early problems came to my attention, or our attention, around sleep and metabolism. And the first of this was that actually many of the patients, when I was asking them about whether they had abnormal movements, would say that they themselves were not aware of it, but their partner who shared the bed with them said that they had movements at night. And that, in fact, led us, when we were trying to quantify chorea with ActiWatches, a precursor of Fitbits and such like, which people wear on their wrist, to try and work out whether we could actually say how much chorea people had. Once we were alerted to this symptom that patients reported, we went back and noticed that lots of the patients actually had involuntary movements as measured by the ActiWatch overnight. And this was interesting because abnormal movements at night obviously could really indicate some sort of sleep disorder. But what it tended to imply was that if people moved at night, traditionally, those would not be present if you actually were asleep, because all movement disorders, especially in Huntington's disease, are essentially generated. So that implied that people perhaps weren't sleeping as they thought. So that was a clue that sleep in Huntington's disease might not be as good as we thought. Secondly, and linked to this, and you'll see how all this comes together at the end, we also were very interested early on in some of the problems that patients had about navigational issues. And actually, over a number of years, we've studied in quite a lot of detail hippocampal problems or hippocampal tasks in patients with Huntington's disease using the equivalent of a virtual Morris water maze, where people have to find hidden platforms by queues, as well as other more imaginative, if you like, cognitive tasks around spatial memory. And what we found was that patients have very profound hippocampal deficits in early stage Huntington's disease, and even in patients who are pre-manifest. So this implied that actually, if you look a bit more closely based on what people told us, that they moved at night in bed, which suggested that they may have problems with sleep at night, so sleep might not be normal, that this may in some ways relate to problems in the hippocampus, and sleep and hippocampal deficits have been linked, and we certainly found that there were hippocampal deficits. And then the other feature which became notable to us, which is well known in Huntington's disease, is the weight loss, which is a major problem in many patients with Huntington's disease. In fact, it can precede the onset of the condition in more obvious fashions. And so many years ago, we looked at this in people with manifest disease, doing metabonomics, where we looked at the profile of serum metabolites in the blood of both patients and transgenic mice. And in manifest patients, we found that there appeared to be a trend towards a catabolic response. In other words, patients tended to be breaking things down in the absence of any obvious reason, which would mean that that would account for their weight loss. So these early observations led us to believe that actually sleep and metabolism may not be as normal as perhaps we thought in Huntington's disease, or at least not simply explained by the abnormal movements or depression and such like, which often goes with this condition. So this led us on to a study which we began now nearly 15 years ago, where we set about trying to look at this in more detail. And so this was a study where we took patients with pre-manifest Huntington's disease. So these are people who carry the gene but don't have any overt features. We also collected people who did not have the gene and were healthy volunteers who were well matched in terms of age and gender. And this was led initially by Anna Goodman, who's down on the bottom there, and was then taken over by Alpar Lazar, who then completed this study, and we published the first results from this in 2015, which tells you basically what I'm going to now run you through, that was sleep deficits were seen in pre-manifest Huntington's disease to a degree that was somewhat alarming, if you like, but that there were no metabolic defects that we could detect in this group at this particular stage of the disease. So as I say, the trial involved this number of pre-manifest and matched controls, and essentially what we did was we looked at aspects of sleep, and this was done through actigraphy. So this is essentially wearing an ActiWatch, a Fitbit if you like, for 14 days, and that records activity during the day and the night. That doesn't tell you about sleep, it simply tells you about activity, so whether people are moving or not. At the end of that 2 weeks of collecting the actigraphy, they came into hospital and they had 2 days of formal polysomnography. So they were in a sleep clinic with EEG electrodes and all the various other devices that were connected up to them. And they had one night to acclimatise, and then we collected data on the second night. Coupled to this, we did a free living metabolic study, where people took heavy water, and we measured actually what their metabolism was in the free living, i.e. when they're at home living life normally. And then they came in for a 48-hour calorimetry experiment where they were locked in a room, and everything that was given to them and everything they expired was measured. And we've subsequently repeated this to see how these events change over time. And at the end, I'll tell you where we've got to with respect to that. So this was set up by Anna and was taken on by Alpar, and there were many others who helped out, particularly Francesca Panin. And all of these times that they came in, they had the standard neurological assessment with the Unified Huntington's Disease Rating Scale. We did a whole variety of cognitive tests, measures of affect. We also did at the baseline body composition, and we also collected routine bloods, including for a whole series of hormones. And what we found was that here we have the sort of basic demographic data showing that the BMI of these patients actually was just within the normal range, more or less, slightly high for the pre-HD, that they were not depressed. So on the Beck Depression Inventory, that was normal. CAG repeat length in our pre-manifest was as you would expect. And they really didn't have any features of Huntington's disease because the total UHDRS motor score was less than 1. So those were the demographics. We then did the metabolic study, And what we found was that actually, if you looked at all of the various different parameters, there were no significant changes in the main hormone levels between the pre-manifest and the controls. In this slide, you can see that we've also split the pre-manifest into those two groups, one close to disease onset as predicted by various models, and the second far from disease onset. Nowadays, we would tend to use more of a disease burden score, and I'll come on to show that was actually more useful in this study. So fundamentally, there were no major changes in the main hormones in these patients. And there were no major metabolic change seen at rest or with activity. So when we looked at all the different parameters that we collected through our calorimetry and our free living metabolism, there was no difference in metabolism between the patients and the controls, which was slightly different to an earlier study where we had shown, as I said, that patients were pre-catabolic, and also in early manifest disease, there was a tendency for patients to have higher metabolism. So at least at this stage of the disease, there appeared to be no major metabolic differences. So accounting for the weight loss in Huntington's disease, we still have yet to attribute. There were a couple of interesting observations which we have no real explanation. One was that they had a tendency for reduced bone mass. So that is plotted up the top there. Quite the reason for that is unclear. And also when we actually looked at energy expenditure as a function of disease burden score, the higher that is, the more you're into Huntington's disease itself, there appeared to be less energy expenditure with increased disease burden score, slightly against what you might expect, given you would expect that if they have weight loss, you'd have increased energy expenditure. But there was no overall abnormalities. It was just a tendency, and that might be reflecting actually apathy, which is also a very common feature of early Huntington's disease. So I think we can say that at least in the pre-manifest stages, we don't pick up major metabolic differences. There is some suspicion that things are not quite right around reduced body mass, but quite like that means we don't know. Once people come into manifest disease, weight loss is more obvious. Whether that has a different basis and there is a sort of catabolic problem there, I think is yet to be determined. So what about the sleep problems? What sleep problems did we see in this group of people with Huntington's disease? Well, quite interestingly, we saw actually quite a number of problems which were quite subtle, but the more you delved into them, the more abnormal they became. So the first observation was that if you simply look at people's sleep diaries, so when they actually record when they go to bed and when they get up, there was actually a tendency for patients with Huntington's disease gene carriers, so these are pre-manifest, don't have the disease itself, to actually go to bed earlier and to get up later. So they spent more time in bed, and that correlated with time to disease onset, i.e. the closer you were, or the closer you were predicted to disease onset, the more time you actually spent in bed. And that was confirmed on our polysomnography. If you actually looked at time of lights out to waking up, that also seemed to be the case. So that's interesting because it implies that people with pre-manifest HD, for whatever reason, tend to spend longer in bed. So the next question was, well, what actually happened when we did polysomnography? So the sleep diary simply told us that patients spent longer in bed. The actigraphy suggested that there was slightly more movement in patients in bed at night, but nothing particularly striking. But what we found when we looked at the polysomnography was that the patients, when you actually looked at them, had a more disorganized sleep. So if you look at REM versus different stages of slow wave sleep and sleep efficiency, you found that actually patients with pre-manifest disease actually had a much more disruptive sleep architecture than you would expect. So superficially, if you look at these patients' polysomnography, there isn't anything particularly striking. But once you start delving into it and looking at it in terms of the different stages of sleep and how it's regulated between those stages, it becomes much less organized. And that this actually becomes more of a problem and more disorganized as patients move closer to the estimated time of disease onset. In other words, as the disease burden score increases, which implies that you're moving more towards manifest disease, the problems with sleep architecture become more florid. So then the question is, well, how real is that, or is that just an artifact? So Alpard did some very more complicated, detailed analysis of electrical activity. So actually looking at the EEG rather than the stages of sleep, and doing some QEEG analysis of patients with the gene and those without. And what he found was that actually, if you look at the spectral profile of patients with pre-manifest Huntington's disease versus healthy controls, they are very abnormal as patients progress closer towards disease onset. So all of this implies that people with Huntington's disease don't really report major sleep problems. This is in the pre-manifest stage. But if you start looking at it, they do seem to have some abnormalities which can be detected rather superficially with sleep diaries. Polysomnography reveals that there are abnormalities around sleep stages and organisation of sleep. And QEEG reveals that there are actually major problems across the spectral profile that you see on the EEG, all of which implies that actually sleep is starting to fall apart as one of the very earliest abnormalities, not in terms of whether people are awake or asleep, but in terms of the way it's organised. And organisation of sleep is obviously very important for healthy brain function, and particularly for cognition. And interestingly, when we looked at the cognitive deficits in our patients, they coincided with these abnormalities that we detected on EEG and polysomnography. In other words, sleep abnormalities and cognitive abnormalities go hand in hand. Now, whether one drives the other is, of course, unknown, but it's rather suggestive that these two are related. So to conclude this talk, what I would say is that weight loss is seen in Huntington's disease, coming to the metabolism aspect of the talk, but the origin of this, I should say, is not fully resolved. And we couldn't find any problems ourselves when we did a fairly detailed, in-depth study of pre-manifest patients. But in manifest patients, we have found other abnormalities. So weight loss is an issue. Quite what underlies it, we don't know. But it's certainly, there isn't a major problem with metabolism, at least in pre-manifest disease. Sleep problems, on the other hand, we think begin very early in the disease course, and they in themselves may give us a better measure of when the disease process has actually begun. So polysomnography may be a way of detecting disease onset, and that, as I say, links to cognitive deficits, and especially with hippocampal deficits, which we've also found to be highly useful in predicting when the disease appears to have started. Problems with sleep may be part of a much bigger circadian type problem, and I think the talk after this will pick up on clock genes and circadian rhythms as to whether this is a more generic problem in Huntington's disease, of which we've only looked at one aspect, namely sleep. Interestingly, of course, if you sleep badly or your sleep is disorganized, as you would with jet lag in the days when we used to fly around the world, that could account possibly for some of the early symptoms and features of Huntington's disease. So if you're tired and jet lagged because you have poor sleep, you tend to be a bit apathetic, you tend to be a bit irritable, you have some cognitive deficits, especially executive deficits. So it could be that many of the early features of Huntington's disease are nothing more than a manifestation of disorganized sleep. Ergo, if you put people back to sleep better, you may actually be able to restore many of the features of Huntington's disease, even if you haven't affected the disease process. Although, interestingly, abnormalities in sleep have often been associated, or have more recently been associated, with lots of processes in the brain. And it is therefore conceivable that if you don't sleep properly, it may drive the disease process through changes in autophagy, lymphatic drainage, and such like. So it is possible that these very early sleep abnormalities are of use in terms of diagnosis, linking to cognitive deficits. They may account for some of the symptoms. But if you can in some way interfere with that and put it back to normal, you may not only make the patient feel better, but you may actually have an effect on the disease process itself. And that has been shown recently in some papers which have suggested that this might be possible. So where are we going now with our data? This is all historical publications data, which we talked about. Obviously, we followed these patients over time. And so Zana Voisey, who's the new clinical fellow who's joined our group, is now looking at the data on these patients that we originally recruited in 2007 and formed the paper of 2015. We've now collected longitudinal data with this. We're now following these patients up, subject to us getting them back into the hospital post-COVID to do a whole series of measures, which we did at the beginning. And the value of this will be, does it tell us that the very earliest abnormalities, which we detected now 14 years ago, are predictive of where patients will be a decade and a half later? And linked to this is the idea that we might now start to try and interfere with sleep early in the disease course, with the hope that we might be able to reverse some of the deficits which we've described over time in patients with pre-manifest Huntington's disease. So I'd like to thank you for listening to this talk this afternoon. Our work has been supported over the years by the National Institute of Health Research here in Cambridge, the Wellcome Clinical Research Facility, where we do a lot of this work. CHDI have been very generous in supporting this work, and ZANA is in receipt of an Association of British Neurologists and Brain Fellowship, and we've had many wonderful collaborators who've worked on this, some of whose work I've not talked about today. So thank you very much.
Yes, thank you very much. Can you hear me? Yeah. So, Roger, thank you very much for this excellent presentation of very important and clinically very relevant work, and also for offering me a great transition with the allusion to the clock gene genetics to